In this video, I am going to give an idea about genotype, phenotype, phenotypic expression, and genetic variation and causes of genetic variation or genetic recombination. So let's start with genotype. What is genotype? It is the sum total of genes that make up the genetic apparatus of a cell, or it is known as genome. And there are two characters or two features for this genotype. These are hereditary constitution of the cell. And what is meant by that? It can transmit this genetic genome or genetic material to the next generation in that form or in a recombinant form when they are fertilized or they are combined with another cell. It may be changed. So that is known as genotype. One more, it will not change with the changing environment. Next one is phenotype. It is the expression of the genotype in a given environment. And one more thing, it will change with the change in environment, and it refers the entire physical, biochemical and physiological makeup as determined both genetically as well as environmentally or phenotypically. So that is the difference between genotype and phenotype. Genotype will not change with the environment and it will transmit it to the next progeny. Phenotype, it is the expression of genes with environment. It will change with the environment and it will not transmit to the next progeny or generation. Now phenotypic expression. This is the expression of phenotype with different situation. Okay, environment മാറുന്നതിനനുസരിച്ചുണ്ടാവുന്ന ചേഞ്ചസുകളാണ് phenotypic expressions എന്ന് പറയുന്നത്. Examples are usually typhoid bacillus are flagellated. They are normally flagellated, but if you are growing this typhoid bacillus in phenol agar, you do not grow flagellae. So flagellae are lost in physical variation. Now, second example is lactose fermentation of E. coli depend on beta-lactosidase enzyme. Okay, in order to ferment the lactose or the E. coli will produce the beta-lactosidase enzyme only in the presence of lactose. In absence of lactose, it will not produce this enzyme. So these enzymes which are produced or which are induced by presence of a substrate is known as inducer enzyme, or the enzymes which are present irrespective or presence or absence of enzyme is known as constitutive enzyme. And this phenotypic variation can be influenced by environmental, limited in range by genes which are not heritable, which will not transmit to the next generation. Now, genotype. Phenotype we already discussed. It is the expression in physical change or environment of the genotype. Now, there is no change will be there unless and otherwise there is a recombination of gene in genotypes. It is the sum total of gene in a cell. It will not change its genetic constitution unless and until it is combined with the genetic material of the another cell. Now, how can this genetic variation can be occurred? Usually genotype variations are stable and heritable, which are not influenced by the environment. It may be changed due to the alteration in the genome. How can alteration can be occurred? There are two methods for the alteration of gene in genotype. That is, first one is mutation and second one is gene transfer. Mutation is the alteration in some point of genotype in undirected way. There is no combination of gene with another cell. So that is the mutation. And in case of gene transfer or recombination, it is the transfer of gene from one cell to another cell. There are different mechanisms for genetic recombination. That one is transformation, conjugation, transduction, protoplast fusion, in vitro combination, genetic recombination method, etc. are the different method for gene transfer or genetic recombination. We should not confuse with the word recombinant DNA technology. Recombinant DNA technology is the method, biotechnological method for gene cloning, in which we will isolate some part of gene and we will combine with some vector, and it will transmit it to an host cell for an predetermined goal. But here it is a just recombination. In case of genetic recombination or gene transfer method, it is just combination of different genetic material from another cell. So that is the main difference between recombination methods or gene transfer method with recombinant DNA technology. Now, first method for genetic variation is mutation. Now let's discuss about mutation. What is mutation? Mutations are random, undirected, heritable variation caused by alteration in the nucleotide sequence at some point of DNA, which occur due to addition, deletion, or substitution of one or more base. Now, mutation can be of two types, which are spontaneous or induced. From the word itself, we can come to know that spontaneous is it will naturally occurring, and inducing, how we have to induce that type of mutation. Now, spontaneous mutation caused by error in DNA replication or by exposure of organism to the natural extracellular influences such as radiation or chemical environments. Now, induced mutation, which are caused by deliberate exposure of organism to the natural extracellular influences, and it can be induced by some substance or some radiation which is known as mutagens. The mutagens are the agent which is used to induce the mutation, and which are of two types: radiations like X-ray, gamma rays and neutrons, and some kind of chemical agents like alkylating agents, acridine dye, bromouracil, 2-aminopurines are example for mutagens. Now, coming to the molecular basis of mutation, it is of two types. First one is base pair substitution, and second one is frame shift mutation. First one, base pair substitution. Spontaneous base pair substitution means because of some abnormal base pairing between the bases of DNA molecules due to the existing of tautomeric form of purine and pyrimidine base in the DNA. There will be a substitution of one purine with another purine or one purine with another pyrimidine. According to that, the base pair substitution is again divided into transition and translation. Transitions are the substitution of purine by another purine molecules and or pyrimidine by another pyrimidine molecule. Let's discuss about the examples for transition. Adenine and thymine are nucleotides which are replaced with the guanine and cytosine are example for transition. One purine is substituted by another purine, and pyrimidine by another purine, guanine, cytosine to adenine. Likewise, purine is replaced with purine, adenine is replaced, pyrimidine is replaced with the pyrimidines. Next one is transvasion, in which purine are substituted with another pyrimidine. Okay, purines are substituted with another purine and vice versa. Okay, next one is induced base pair mutation. By structurally similar purines or pyrimidine, we can induce the base pair substitution. That is, base analogues and deaminating agents can be used to induce the base pair substitution. Now, second one is frame shift mutation. It can be done by, or it can be induced or naturally by deletion, addition of one or more nucleotide. That is the molecular basis of mutation. Now, coming to the consequences of point mutation. first one is missense mutation. Okay, so mutation that involved at the single base pair substitution in the DNA maybe leads to substitution of one amino acid for another. Usually it will give one amino acid by changing one nucleotide of the three coded codons. It will change to another amino acid, that is known as missense. Our sense will be changed from one amino acid to another amino acid, so that is known as missense mutation. For example, code AAU is in AAU there is a mutation occur at uracil and it is changed to AAG. In that what will happen, we have to get asparagine as an amino acid, but after the mutation we will get lysine. Okay, അങ്ങനെ ഒരു ചേഞ്ച് ഉണ്ടാവും എന്നുള്ളതാണ് മിസ്സെൻസ് മ്യൂട്ടേഷൻ എന്ന് പറയുന്നത്. സെക്കൻഡ് വൺ ഇസ് നോൺസെൻസ് മ്യൂട്ടേഷൻ, in which alteration in the base sequence of the DNA resulting into the formation of a codon which does not code for amino acid. That means we have three type of nonsense അല്ലെങ്കിൽ nonsense codons are there, in which it will give a punctuation mark. So that will occur in nonsense mutation, which will not code for any one amino acid. After the mutation, this code will change to code which will not produce any kind of amino acid. That is known as nonsense mutation. Now, suppressor mutation is a reversal of mutant phenotype by another mutation at the point of DNA distant from that of original mutation. Same in this mutation do not occur in the same point, but it is some distance apart from the first mutation, but it will not change the amino acid sequence. It will reverse the first mutation by the second mutation. That is known as suppressor mutation. And next one is silent mutation. In silent mutation, substitution of one nucleotide base pair for another may not lead to the change in amino acid. We means after the mutation it will produce a new codon which will represent the same amino acid as that of the previous one. That is known as silent mutation. And last one is lethal mutation. After this type of mutation, it will result into the permanent death of microorganism, which will make into unable to reproduce. So that is known as lethal mutation. So it will kill the cell of cell, and this is known as lethal mutation. So this is the pictorial representation of missense mutation. See here we can see the code UAA. GC is the code for amino acid glycine. After the mutation at the cytosine, it will change to thymine, and complementary adenine will be formed, so that instead of getting methylalanine, lysine, phenylalanine, we will get methylalanine, lysine, phenyl and serine type of amino acid. We will get it will completely change the sense of that amino acid sequence. We know the proteins are sequence of amino acid in a specific way. Each protein have its own amino acid sequence. So by changing even a single amino acid, it will completely change the nature of that protein. So that is it will cause the missense of that proteins. And second one is nonsense mutation. After in case of this, you see TTC and its complementary AAG is representing for the lysine. After the translation, it will represent for the lysine. By changing the site at instead of thymine, it will change to adenine. It will lead to an codon which represent full stop codon or nonsense codon. So it will break the chain of that amino acid sequence there itself. And last one is frame shift mutation due to the deletion of adenine from this side. It will change the amino acid sequence to methylalanine, lysine, phenylalanine to serine to methylalanine, lysine, and alanine. So that is the frame shift mutation. So this is all about the mutation. Now let's go for the second fact or second method for genetic variation, that is recombination methods or gene transfer methods are the another method for genetic variation. Now first let's go for the what are the different mechanism of gene transfer or genetic recombination method. First one is transformation, conjugation, transduction, protoplast fusion, electroporation, lipofection, and in vitro genetic recombination or direct recombination method are the different methods for gene transfer method or genetic recombination methods. Now first one, transformation. All this method are transfer in this method there will be a transfer of gene from one cell to another cell. Only difference is in which way they are transferred. Either it is as a naked DNA or free DNA ആയിട്ടാണോ, or through a cytoplasmic bridge, or through an vector, or through artificial method. That is the difference here coming. First one is transformation. So it is defined as the transfer of genetic information through an agency of free DNA through its environment as a naked soluble DNA. So if we are mixing two cells in a solution, genetic material of one cell will go to another cell through that solution through an environment as a free naked DNA. That is the difference in with another gene transfer method, transformation method. Actually, this method has discovered by an experiment conducted by Griffithing. He conducted an experiment with mice in which he injected a mixture of live non-capsulated rough strains of pneumococci, which are non-virulent, which will not cause a pneumococcal infection and which will not kill the microorganism. And he mixed this live non-capsulated rough strains of pneumococci with heat-killed capsulated pneumococci. Heat-killed capsulated pneumococci also do not kill the mice because he already destroyed the infectious nature of the live heat killed capsulated. He already destroyed that nature, disinfection nature or infection nature of pneumococcal infection. So when he mixed both the strains in an form in which it will not cause the infection, it will not cause the death of microorganism, but when he mixed this one, the mice got dead. So from that he came to know that, so he conducted an experiment in such a way that first he injected smooth strains smooth strains of virulent pneumococcal infection in live form. When he injected to the mice got dead. And when he injected the rough strains, non-capsulated strains to the mice, mice didn't got infection and mice alive, it didn't die. Now he killed the smooth strains or pathogenic bacteria and injected, mice didn't kill. But when after he mixing the harmless and heat killed pathogenic bacteria, when injected, the mice dead. From this experiment he concluded that even though both the mixture of live harmless bacteria as well as heat killed bacteria will not kill the mice individually, when he mixed, something happened in that mixture. That means some property has been transferred from the heat killed pathogenic bacteria to live harmless bacteria. So from this he concluded that there will be a transfer of gene through an environment, so that is known as transformation. So in order to perform the transformation in laboratory, we can follow this step, that is uptake of plasmid DNA by E. coli is carried out by ice cold calcium chloride 0 to 5 degrees Celsius and subsequent heat shock 37 to 45 degrees Celsius about 90 seconds. And this transfer was carried out by the calcium chloride, which will affect the cell wall break at the localized region and is also responsible for the binding of DNA into the cell structure. And a brief heat shock stimulate the DNA reuptake also. So that is the transformation. So next one is conjugation. So it is the process of direct transfer whereby donor cell or male cell make a physical contact or cytoplasmic bridge or conjugation tube with another cell or recipient or female cell. So it will transfer genetic material from one cell to another cell through an cytoplasmic bridge, which is known as conjugation tube, which are produced by donor cell or male cell with the recipient cell. So inside the recipient cell, the new cell may integrate with the chromosome or may remain free. Conjugation occurs among the cell from different genera of bacteria like Salmonella and Shigella. The maleness of the donor status is determined the a plasmid which is known as F plasmids or plasmid responsible for the production of conjugation tube is known as F fertility factor. The cell which have fertility factor is known as male cell or donor cell, which do not have is known as female cell or recipient cell. So this is the plasmid which is responsible for the conjugation or production of conjugation tube or cytoplasmic bridge. So this is the method of gene transfer. Conjugation through an cytoplasmic bridge is known as conjugation. Now next one is transduction. Transduction is defined as the transfer of portion of DNA from one bacteria to another bacteria by an bacteriophage or vector through an vector which will carry genetic material from one cell to another cell is known as transduction. So we already had studied about the different life cycle of bacteriophage. There are two types of life cycle, lytic cycle and lysogenic cycle, in which bacteria will take some part of one's genetic material of one cell to another cell by infection or replication of genetic material. So there are two types of transduction: generalized transduction, which will involve any segment of DNA at random, and restricted transduction when specific bacteriophage transduce only a particular gene trait. And transduction affect the plasmid and episomes. Plasmid transfer include penicillin resistance in Staphylococcus, and which will help in genetic mapping also in eukaryotic cells. So there are two types of bacteriophage. Bacteriophage, we already know it is a complex type of virus which usually used to replicate by using another cell, usually E. coli or another bacterial cell. So while replication of these bacteriophages, it will take some part of gene from one bacteria, and while attacking the next bacteria, it will transfer the genetic material which was achieved or which was taken in previous replication process. There are two types of lytic cycles of transduction, that is lytic and lysogenic cycle, or lytic or virulent cycle, in which after a large number of progeny are built inside a host bacterium and which will rupture the bacteria and phage is released to attack next cells. And second one is temperate or non-lytic cycles, or host bacterium is unharmed. It will be there in that bacterial cell unless and otherwise some stimulus has been released to rupture the bacteria and to coming for attack of next type of bacteria, that is the lytic and lysogenic cycles. Next one is protoplast fusion. So what is protoplast? A cell without cell wall is known as protoplast. So in this method we are artificially fusing two protoplast in order to transfer the genetic material. So recombination between two different species of strains can be enhanced through these techniques. This is employed to both for plant as well as animal cell. An isolated protoplast, that is cell wall, can be digested by enzyme. Enveloped by membrane only can be produced in a medium which is isotonic with the content. In laboratory, usually for performing this protoplast fusion, we will use polyethylene glycol, or by using pulsed electric field, we can enhance the uh, uh, protoplast fusion, which will induce the fusion of protoplast. Next method for gene transfer is electroporation. This method based on the principle that high voltage electric pulse can be induced cell plasma membrane to fuse. This technique involves electric field mediated membrane permeation. Electric shock also induces the cellular uptake of exogenous DNA, which is believed to via the pores formed by electric pulses from the suspending solution. Usually what we have to do, the cell placed in a solution containing DNA and subjected to an electric shock to cause holes in the membrane. The foreign DNA fragment enter through the hole into the cytoplasm and they will recombine with this one. This is a pictorial representation of electroporation. We have to take the cells and we have to take the DNA fragment which have to be transferred to the cell in a vessel in which we are mixing, and we have to keep it. This is a pictorial representation of an electroporation in which the cell in which to the DNA has to be transferred, as well as DNA fragment which to be transferred are mixed in a solution or in a special cuvettes in which we will keep the optimum temperature for that one, and we will give an electric pulses, so it will form the pores on the cell membrane which will cause the permeation of DNA fragment to the cells, and which will fuse with the which will enter into the nucleus of the cells. This is a method of electroporation. This is another method for gene transfer from one cell to another cell. Next method is lipofection or liposome mediated gene transfer. As we all know, all the materials will cross the membrane unless and otherwise it is an lipid membrane. Based on this principle, we are using lipid molecules which have an aqueous. Liposomes are circular lipid molecules which have an aqueous interior that can carry nucleic acid. The liposome mediated gene transfer is known as a liposome. On treatment, the DNA fragment with the liposome, the DNA pieces get encapsulated inside the liposome. The liposome can adhere to the cell membrane and fuse with them to transfer the DNA fragments. Thus, the DNA enter to the cell and then it will fuse with the cell which has to be transferred. So this is the pictorial representation of liposome and DNA fragment. We are mixing with the DNA, it will encloses the liposome mediated membrane along or around the DNA fragments, and after adding to the cells, it will fuse with the cell into which it is to be transferred. Since it is a lipoidal membrane, it will fuse and it will transfer the DNA fragment to be transferred to the cell, and it will enter to the nucleus of the DNA. That is liposome mediated gene transfer methods. And last one is in vitro genetic recombination or direct transfer of DNA methods. It can be done by micro injection using that kind of needles used for culturing of cells, used to introduce DNA into large cells such as oospores, egg cells. Egg and cells of early embryos are used to transfer this type of genetic material. And last one is particle bombardment. Now we are bombarding two cells in high speed. It will transfer the genetic material from one cell to another cell through particle bombardment method. So these are the different methods for genetic variations. So in this video we already discussed about the genotype, phenotype expression, and genetic variation, and what are the different methods for genetic variation. First one was mutation and second one was gene transfer of genetic recombination method. In mutation there is some error occurred due to the replication, that is the only change in the genetic material. But in case of gene transfer method or recombination method, in this genetic material is transferred from one cell to another cell through some mechanisms like conjugation, transformation, transduction, protoplast fusion, lipofection, or in vitro methods. So these are all about genetic variation. Genetic recombination methods is an important topic for your exam, so please study as important question. Thank you all.